pakitawag na. Magsisimula tayo in 4 minutes. Thank you. Mhm. 10 minutes, 2:10 na. So, mag-start na tayo. Ilan pa lang kayo? 35. Yung ibang classmates nyo, nasan sila? Ayun. So, actually, tinitingnan ko pa rin yung attendance kahit na hindi ako nagtatawag. Meron kasi sa Google Meet na automatic na check yung attendance. Pero yung attendance nyo naman, nakabase rin yun sa class participation forms. So, dapat mag-fill out pa rin kayo ng class participation forms nyo para makount ko yung attendance nyo. Okay? So, good afternoon ulit, class. Naririnig nyo ba ulit ako? Pa-confirm lang.
Yes.
Yes po.
Okay. So, naririnig naman. Ngayong hapon, yung i-discuss natin ay tungkol sa cell culture. Ayun, cell culture. Wala na akong ibang malagay. Yan lang na meme na yan. And, kaya natin naaralin yung cell culture kasi isa rin siya sa mga basic laboratory techniques na kailangan yung matutunan na pwede niyong magamit balang araw as medical technologists. And yung cell culture ay parang siya rin yung marami siyang applications in medicine. Ang kailangan nating matutunan ngayong hapon ay yung basic principles of cell culture. So, yung lecture natin ngayong hapon, medyo maikli lang. Mabilis lang tayo matatapos, I think. Maikli lang yung ginawa kong PowerPoint. Mga 40 slides lang to. So, ayun. Simulan natin. First, titingnan natin yung history ng cell culture. Next, titingnan din natin yung cell culture categories or yung types ng cell culture. And tingnan din natin yung cell growth conditions, kung ano ba yung conditions ng cell para makapag-grow sila in a culture. And titingnan din natin yung laboratory setup for cell culture. Tanong lang muna, ano sa tingin nyo, pag nung narinig nyo yung word na cell culture, ano yung parang pumasok sa isip nyo? Hello, meron bang gusto magsabi or mag-recite? Meron bang gusto mag-recite? Yung narinig niyo yung word na cell culture, ano yung parang unang pumasok sa isip niyo? Yes, Queen. Sinis,
Ano po, ay, gaya po doon sa bakte po, mag-isolate po, for example, ng cell, tas ilalagay po natin siya sa culture medium, tapos ipapagrow po natin siya in a sense na, ano po siya, hindi po yun yung natural environment niya.
Tama. So, ayun. Narinig namin yung kapatid mo ba yun? Ayun, tama yung sinabi ni
Sorry
Queen.
po.
So, tama yung sinabi ni Green. Sabi niya, yung cells daw, parang sa bacteriology lang rin or sa bacteria. Basically, lalagay mo sa isang medium tapos papatubuin mo doon. I-isolate mo muna tapos papatubuin mo sa isang medium. Ang kaibahan lang dito sa cell culture na gagawin natin, ang cells na papatubuin natin ay from eukaryotic cells. So, pwedeng gumamit from humans, pwede rin gumamit from animals, and pwede rin na gamitin natin ay plant cells. Pero, yung ifo-focus natin ay yung pag-cell culture ng um, eukaryotic cells ng mga humans or ng animal cells. Kasi yun yung parang pwede nyo gawin sa laboratory nyo balang araw. Ayun. Pero pwede ka rin magpatubo actually ng cell culture using plant cells. Tapos pwede kang gumawa ng or humanap ng secondary metabolites. Usually, yung mga secondary metabolites sa plant cells, ginagamit sila as gamot. Ayun. So, history muna. Konting history. Wait lang. Nagugulo yung slides ko. Asan yun? Ayun. So, nung 1670s, nagulo yung slide ko. Nung 1670s, parang nagsimula yung study sa cell biology. Paano? Si Anthony van Leeuwenhoek, siya yung parang pioneer ng microscopy. And ginawa niya, gumamit siya ng glass, tapos parang gumawa siya ng glass kung saan pwede niyang gamitin as a microscope. And tiningnan niya yung um, yung tinga niya actually. Yung tinga niya, galing sa ngipin niya, tiningnan niya, tapos nakita niya doon sa tinga niya, merong mga bacteria, merong gumagalaw-galaw na maliliit na mga bagay. And yung nakita niya, dinrawing niya dito, nakita niya to, figure 8B, CD, nakita niya, yan yung nakita niya, mga gumagalaw-galaw na small organisms. And tinawag niya yon na animalcules. So sabi niya, meron daw diverse animalcules siyang nakita using yung simple microscope niya. And also, kumuha rin siya ng water from pond. And nakita niya rin yung volvox. Itong figure 2, kung nakikita niyo man sa PowerPoint, yan yung drawing niya ng volvox. So from then on, nakapag-develop na or nagkaroon ng development sa cell biology. And Ngayong 21st century, ngayong 21st century, ang dami na nating advancements in cell biology. Isa na doon ay yung FRET or Forster Resonance Energy Transfer na pinakita natin doon, di ba? Nung nag-aral tayo ng DNA damage and repair. Yung FRET, dinevelop siya ni Theodore Foster para magkaroon ng kulay or label yung DNA para makita niya yung processes doon sa cells. And also, yung pinakasikat na lab technique which is PCR or polymerase chain reaction, ang nag-develop nun ay si Carrie Mullis. Si Carrie Mullis, chika lang, na-develop niya yung PCR actually, nasa kotse siya kasama yung girlfriend niya. Tapos bigla niyang naisip, gumamit siya ng different temperatures para ma-amplify yung DNA. Yun yung parang history kung paano na-develop ni Carrie Mullis yung PCR. So, ayun. Sana all, di ba? Parang nasa kotse ka lang kasama yung jowa mo. Meron ka lang na-develop na advancement in molecular biology or in cell biology or lab technique, di ba? So, yun. And sadly, si Carrie Mullis, nag-pass away siya nung 2019 lang. Ayun. So, isa siyang pinakakilala or sikat sa field ng molecular biology kasi bago magkaroon ng PCR, walang way or hindi ganon ka-specific yung way to detect DNA sequences. And sobrang laki or sobrang dami ng applications ng PCR. So, ayun. So, ngayon, sa 21st century, in our modern days, marami tayong new methods for analyzing cells, proteins, DNA, and RNA. And marami rin tayong tinatawag na molecular blueprints. And kaya na natin itong ma-access sa online. So we have an access to sequences of many billions of nucleotides, proving the complete molecular blueprints for hundreds of organisms. So anong implications nito? Sabi daw, marami na naka-database yung blueprints ng sequences ng many organisms. Anong implications nun? Marami siyang implications. Kung alam mo yung sequences ng different types of organisms and yung genome nila, pwede mo rin aralin yung specific genes. Kung nyari may interest ka sa isang genes, pwede mo yung aralin and in turn, aralin mo yung protein na pinoproduce ng gene na yon. And kapag inaral mo yung protein na pinoproduce ng gene na yon, pwede mo nang maintindihan ngayon yung processes ng cells natin. Or pwede kang ngayon mag-develop ng mga gamot targeting the proteins or yung mga genes na yon. So, from 2002 to present, nag-boom yung total bases or sequences, yung number of bases sa loob ng GenBank. GenBank yung tawag sa online database ng mga genome sequences ng halos lahat ng organism. So, kahit ngayon, i-search nyo yung GenBank, NIH, GenBank, i-search nyo yung GenBank, tapos mag-search lang kayo, kunyari, sequence ng E. coli or sequence ng Salmonella. Nandun yon or sequence ng kunyari protein na nasa rat. Nandun sila lahat. So, pwede na natin ngayon ma-access yung mga sequences na to and maaral natin. And bakit to connected sa cell culture? Kasi in cell culture actually, yung pinaka-goal in cell culture is to produce certain types of proteins or to observe certain types of proteins na makikita natin mamaya. Yan. So, in 1907 din, dito rin parang nagsimula yung cell culture or tissue culture. Nagsimula yung cell culture, basically, medyo funny para sa akin kasi nag-aaway yung mga scientists noong 1907 tungkol sa neuronal doctrine. Yung neuronal doctrine or mayroon kasing controversy sa neurobiology, pinag-aawayan nila kung yung nerve fiber daw ay parang yung growth niya daw from one cell lang ba or nagdi-divide sila into many cells ganon. So, nakita nila na yung neurons each nerve fiber is the outgrowth of a single nerve cell and not the product of the fusion of many cells. So, yung pinag-aaway nila pala actually ay kung yung isang nerve cell ay isang buong cell o yung nerve cell ay parang pinag-connect-connect na different types of cells. Kasi 'di ba weird yung itsura ng nerve cells. Parang sa unang tingin mo, akala mo yung central na cell different siya doon sa outgrowth niya, dun sa mga lines. So, yun yung pinag-aawayan nila. So, ngayon, itong mga scientists noong 1907, ginawa nila, nag-culture sila ng nerve cells. And doon na parang nagsimula talaga yung cell culture. And ayun nga, nakita nila na yung nerve cell ay galing lang sa isa lang siyang talagang cell. And ito na nga, yung application ng cell culture ay nagamit noong 1948. So, nagkaroon tayo ng polio vaccine production by John Enders and his colleagues. Sila yung pioneers and nanalo pa sila ng Nobel Prize in Physiology or Medicine. Si John Enders at yung colleagues niya, ginawa nila, nag-cell culture sila, tapos sa cell culture na yon, nag-produce sila ng polio, parang weekend polio strain. And yung weekend polio strain na yun yung ginamit nila as vaccine. So, from then on, 1948 pa lang, alam mo na yung cell culture, pwede siyang ma-apply din into vaccine development. So, ayun. And ultimately, kaya rin tayo nagsa-cell culture is to understand what takes place inside the cell. So, kapag gumamit ka ng cell culture o kapag tinignan mo yung cell cultures natin, kapag inaral mo yung cell cultures natin, makikita mo rin na maiintindihan mo rin kung paano nag-work yung inner workings of the cell. Ito na. So, ito ngayon yung cell culture categories or yung different types ng terms na ginagamit sa cell culture. First, let's define what is cell culture. So, cell culture is the removal of cells from an animal or plant and their subsequent growth in a favorable artificial environment. Ano daw? Sabi? So, sabi daw, from an animal or plant, pwede kang magtanggal ng cells and yung cell na yon, pwede mong ipag-grow daw in an artificial environment. And kung nakikita niyo man sa picture, 'di ba? It's under the flow hood. Dito ganito yung itsura ng setup sa flow hood ng 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 cell culture. So ito dito sa flask na to, dito pinapatubo yung cells. Wala pa siyang or meron na tong laman. Kung nakikita niyo man medyo yellowish yung flask. Meron na yang cells. And itong mga reagents na to, yung ginagamit para magpatubo ng cells. And makikita natin mamaya yung different types ng reagents na ginagamit. So, ayun. So again, cell culture is the removal of cells from an animal or plant and their subsequent growth in a favorable artificial environment. So animal or plant daw, 'di ba? So yung usual na alam natin na pwedeng i-cell culture ay yung animal cells or yung galing sa tao. Pero pwede ka rin actually mag-cell culture ng plants. So, ayun. Nung undergrad ko kasi sa biology, nag-cell culture kami ng plants and sobrang hirap mag-cell culture ng plants. Lalo na kung parating tinutubuan ng fungi. So, imbis na developmental biology yung inaaral namin, nagma-mycology kami kasi ang daming fungi nung plant cell culture namin. Yun lang. So, yun yung mga undergrad stories ko nung bio. Ayun. Ito naman yung tinatawag natin primary culture. Primary culture is prepared directly from the tissues and proliferated under the appropriate conditions until confluence is reached. Define muna natin yung define muna natin yung primary culture. So primary culture daw, prepared directly from the tissues and proliferated under the appropriate conditions under confluence is reached. Primary culture, basically, yun yung tissues na una mong nilagay sa cell culture. So, nasa word itself, siya yung una mong tissues na or una mong cells na nilagay doon sa culture mo. And itong primary culture, mag-grow sila until confluence is reached. So, ano daw yung confluence? Yung confluence, meaning lang yan, yung area na kung ilang area na ilang percentage ng surface ng culture dish ang na-covered nung cell culture mo. So, confluence refers to the percentage of the surface of a culture dish that is covered by your cell culture. So, for example dito, meron kang 10% confluence. 10% confluence kasi 10% lang yung nako-cover doon sa medium mo, nung mga pinapag-grow mong cells. Dito naman, 30% confluence kasi 30% lang rin yung nako-cover nung pinapag-grow mong cells. Dito, 50% confluence kasi kalahati na nung medium mo na-cover na nung cells. At dito naman, 90% confluence na kasi 90% na rin yung na-cover doon sa cells mo. So basically, yung confluence again, kung ilang percent na yung na-cover doon sa culture medium mo, nung cells na pinapag-grow mo. And primary culture, they can only divide a finite number of times due to telomere loss. So tandaan nyo, yung primary culture, yung unang cells na pinapag-grow nyo, finite lang daw sila tutubo. Ibig sabihin may limitations lang. Kasi meron tayong telomere loss na tinatawag na inaral na natin last time, di ba? Kapag yung cells nag-divide sila at a certain time or every time na nagde-divide ang cells, umiikli rin yung telomeres nila. And kapag yung telomeres na nila ay sobrang ikli na, there will come a time na yung cells natin mag-undergo ng senescence. And senescence is yung kung saan magsa-stop na mag-grow yung cells natin. Parang matutulog na yung cell. Kasi sobrang ikli na ng telomeres nila at hindi na kayang maproteksyonan ng telomeres yung DNA nung cells na nagde-divide. So therefore, normal cells can only divide a finite number of times, and ito yung primary culture mo rin. Ayun. Yung secondary culture mo naman, sila na yung subculture or passage from your primary culture. So secondary culture, obviously, sila na yung cells na kinuha mo doon sa primary culture tapos pinatubo mo ulit yung primary culture. So yon. Yung term na kailangan yung tandaan ay passage. Yan yung parang term na ginagamit para patubuin yung cells natin or transfer ng cells from one medium to another. Passage yung tawag doon. Ayun. So, magkakaroon tayo ng established cell line kapag yung cells natin, they can grow indefinitely na raw. What do you mean by that? Established na yung cell lines mo, yung cell culture mo, pwede na siyang tawagin na cell line kapag infinitely na nagpo-proliferate na siya, kapag paulit-ulit na siyang nagpo-proliferate kahit pabayaan mo lang. 'Di ba? Parang, ano sa tingin niyo, 'di ba? Parang mga cancer cells, 'di ba? So, these are used in research, particularly in vaccine research. Kanina nakita natin yung poliovirus, yung pag-produce ng polio vaccine, pag-produce ng polio vaccine, ang ginagamit ay through cell culture. And also, cancer research. Sobrang important ng cell culture. And also in protein therapy. Kasi sometimes yung cell culture, pwede rin siyang gamitin to produce some sort of proteins. And, ano nga yung example ko dun? Proteins, katulad ng yung sa, wait lang. Yung sa diabetes, um, yung insulin. So, dati kasi, yung insulin, kailangan mo pa kumuha ng cells mismo ng pigs, cells nila ng pancreas para makakuha ng insulin. Eh, ilang insulin lang ang makukuha mo sa pancreas ng pigs para makakuha ka ng significant amount ng insulin para magamot mo yung diabetes, di ba? So ngayon, nagkaroon tayo ng cell culture and ito na ngayon yung technique na ginagamit para mag-produce ng insulin. And dahil doon, mas marami na tayong or mas marami ng amount ng insulin yung napoproduce and kaya na rin mag-produce ng insulin na kayang makapaggamot sa diabetic patients. Ayun. And established cell lines, they are obtained primarily from clinical tumors. 'Di ba? Kasi, again, yung established cell lines natin, sabi daw, they can grow indefinitely. They can proliferate and proliferate and proliferate. So, ano yung mga cells na proliferate lang ng proliferate, yung tubo lang ng tubo, di ba? Cancer cells. So, therefore, cell lines mo ngayon, galing sila primarily from tumors. And example na pinaka-famous ay yung HeLa cell line, galing kay Henrietta Lacks. Si Henrietta Lacks, 1950s ba siya? Mga 1950s pa yung cells niya. Pero hanggang ngayon, nagagamit pa rin siya ng researchers. So, imagine niyo ilang years na, tumutubo pa rin yung cells ni Henrietta Lacks. At nagagamit pa rin siya sa research, di ba? Mga 70 plus years na siyang patay, pero yung cells niya buhay pa rin. And galing yung HeLa cell line sa tumor niya or sa cancer niya. And also, you can establish a cell line via transforming your primary cells with viral oncogenes or chemical treatments. So, what do we mean by transforming? So, 'di ba yung primary cells natin, sabi natin, finite lang silang tumutubo or meron lang silang limitations kung hanggang kailan lang sila tutubo. Ngayon, dahil may limitations yung kung hanggang kailan lang sila tutubo, pwede mo silang i-transform ng maging cells na indefinitely na tumutubo. So, um, yung primary cells na parang tinanggal or yung primary cells na nag-experiment ka para tumubo sila indefinitely, yung tawag doon ay transformed cells. So, paano natin natatransform yung primary cells into cell lines which can grow indefinitely gamit ang viral oncogenes or chemical treatments. Anong naaalala nyo sa oncogenes? Diba? In the presence of oncogenes, sa presence ng oncogenes, nagkakaroon ng cancer. Diba? Kasi, kailangan mong i-treat yung primary cells ng oncogenes or chemical treatments para ma-induce mo rin yung parang cancer or parang magkaroon ng cancer yung primary cells in a way na kailangan mong i-target yung cell cycle checkpoints. So, transformed cell lines, tinatanggal yung cell cycle checkpoints. So, cell cycle checkpoints are targeted to transform cells. Para makapag-transform ng cell lines, kailangan natin i-target yung cell cycle checkpoints. So, ano ulit itong cell cycle checkpoints? Review ulit natin. So, in cell cycle checkpoints, meron kang, excuse me lang, mabilis ba ako or nakakasunod pa ba kayo or masyado ba akong mabilis? Ibagalan ko ba? Hindi ko kaya nakikita. Medyo bagalan ko ba? Or okay lang? Sige. 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 So, nandito na tayo sa transform cell lines. Okay lang daw. Okay. So, sana nakakasunod kayo sa discussion natin. So, sabi lang kayo, mag-raise lang kayo ng hand kung gusto nyo ulitin ko yung sinasabi ko or may hindi kayo naintindihan. Okay? Mag-chat lang kayo or mag-raise ng hand. Okay? So, transform cell lines, again, yung gagawin mo dito, ito transform mo yung primary cells to cell lines para tumubo sila indefinitely. So, here, you target these cell cycle checkpoints. I-review ulit natin yung cell cycle checkpoints. Meron kong cell cycle checkpoint dito sa GAP1 or sa G1, which dyan tinitingnan kung kaya na mag-commit ng cells para mag-cell division. So, in GAP1 checkpoint, it checks if it can allow the cell to commit cell division. In the S phase naman, anong naaalala nyo sa S phase? Sa S phase, nagkakaroon ng synthesis ng DNA. Sa S phase, dito nagkakaroon na nung DNA replication. Sa S phase, actually, nagkakaroon ng DNA replication. And in the S phase checkpoint, it is where you check the quality of the replicated DNA and if you need to repair your DNA. If you need to see if you need to repair your DNA. Tinitingnan sa S phase cell cycle checkpoint kung may damage yung DNA. And if may damage, mag-activate sila ng repair mechanisms na inaral na natin, di ba, nung nakaraan. So, dito yung nangyayari sa S phase cell cycle checkpoint. Sa GAP2 cell cycle checkpoint naman, halos same lang rin yan sa GAP1 cell cycle checkpoint. Parang ini-ensure lang din nung cells mo kung yung DNA na na-replicate ay tama ba and undamaged na and kung kaya niya na mag-commit to cell division. And dito na nangyayari yung cell division sa mitosis, di ba? So, dito na sa mitosis, iba rin yung cell cycle checkpoint dyan. Titingnan dyan kung tama ba yung pagka-align ng chromosomes. So again, sa GAP1, titingnan kung kaya bang mag-commit ng cells na mag-cell division. Sa S phase naman, tinitingnan kung merong unreplicated or damaged DNA. Sa GAP2, tinitingnan ulit kung merong unreplicated or damaged DNA and kung kaya niya na mag-commit to cell division. And kapag na-check na nung cells mo or nakapagdaan na yung cells mo sa cell cycle checkpoints na to, magpo-proceed na siya to mitosis. And in transform cell lines, yung ginagawa, ito-target mo to cell cycle checkpoints para walang checkpoints. Para mag-cell division lang yung cell mo. Hindi mo na kailangan i-check yung cell kung kaya niya ba mag-cell division. So, yung ginagawa, lalagay ng oncogenes, 'di ba? Yung oncogenes, in the presence of the oncogenes, nagkakaroon ng parang ng cancer, 'di ba? Kasi na-affect yung cell cycle checkpoints. So, ayun. And also, you have to consider din yung tumor suppressor genes. So, kailangan mo rin inactivate yung tumor suppressor genes mo para hindi magkaroon ng cell cycle checkpoints. And ayun, so once the cell cycle control is evaded, the cells continue to proliferate indefinitely. Kapag natanggal mo na tong cell cycle checkpoints dahil wala nang mag-check kung kaya niya bang mag-cell division, edi magsi-cell division lang siya na magsi-cell division and magpo-proliferate lang na magpo-proliferate yung cells mo. At yun na yung tinatawag mo ngayon na cell line. So, ulitin natin. Yung established cell lines mo, they are able to grow indefinitely. And yung kaibahan niya sa primary culture mo, yung primary culture mo, finite lang pa lang yung pag-grow ng cells mo. Pero kapag transform mo yung primary culture mo, kapag transform mo yung primary culture mo into cells na kaya mag-grow indefinitely, they are now called your cell lines. Okay? Ayun, ito. So, merong table, pero di nyo naman yan kayo memorize. Familiarize nyo lang. Yan. Parang, itong table na to, ito yung commonly used cell lines in research. So, makikita nyo itong HeLa cells. Yun yung commonly used na pinakasikat kasi parang pinaka nauna na cell line. Ang HeLa cells ay epithelial cells. And galing ulit siya kay Henrietta Lacks. And hanggang ngayon, tumutubo pa rin yung cells ni Henrietta Lacks. Amazing, di ba? So, cell lines derived from primary cultures have a limited lifespan. And as they are passage again is the transfer of your cell culture from one media to another. Cells with the highest growth capacity predominate, resulting in a degree of genotypic and phenotypic uniformity in the population. So, anong sabi dito? Yung cells daw na may highest growth capacity, sila yung nagpe-predominate. So, para rin tong kung sino yung pinakamalakas, siya rin yung mas marami. So, yung cells na merong highest growth capacity, yun yung kukunin mo para magkaroon ng uniformity in the genotypic and in the genome and the phenotype of your cells. So yun rin yung parang yun rin yung gagamitin mo na cell culture. And yung term naman na cell strain, ginagamit if a subpopulation of a cell line is positively selected from the culture by cloning or some other method. This cell becomes a cell strain. So, sabi daw, kapag yung subpopulation ng cell line mo, nung established cell line mo, nagkaroon ng other or nagkaroon ng other characteristics dito, it acquires additional genetic changes subsequent to the initiation of the parent line. Kapag nagkaroon ng additional genetic changes, yung nauna mong cell line, tinatawag yun na cell strain. Para lang rin, di ba, sa bacteria, may iba-iba tayong strains ng bacteria. Parang ganun lang rin naman yung term dito sa cell culture. Parang same pa rin siya na cells, pero meron lang na onting pagkakaiba sa genome nila. Okay. Ayun. So, meron ba kayong tanong so far? Wala naman. So, proceed na tayo sa cell growth conditions. Dito naman, titingnan natin kung ano yung conditions ng cells para mag-grow sila. So, cells are obviously isolated from tissues. So, but first, you have to disrupt the extracellular matrix and cell-cell conjunction. So, sorry, wala akong nahanap na picture, kaya ang daming words. So, explain ko na lang. Bago tayo makapag-isolate ng cells, kailangan natin i-disrupt yung extracellular matrix and cell-cell junction ng cell tissue, nung tissue natin. Bakit? Di ba kasi yung cells natin, kapag, di ba, sa tissue, dikit-dikit sila. Dikit-dikit yung cells. Kaya paano ka magkukulture ng cells kung magkakadikit sila? So, dapat, yung una mong gagawin, tanggalin mo yung pagkakadikit-dikit nila by disrupting yung extracellular matrix nila and yung cell-cell conjunction nila. Paano? Paano mo sisirain yung pagkakadikit-dikit nila? You use proteolytic enzymes. So, proteolytic enzymes, they break molecules of proteins into peptides. So, from sa word niya na itself. Kaya nilang ma-break yung proteins sa extracellular matrix and cell-cell junction, um, and they eventually break molecules of proteins into peptides and eventually into amino acids. And proteolytic enzymes are used to digest proteins in the extracellular matrix. Inuunahan ko na sarili ko. Ano ba yan? So, example ng proteolytic enzymes ay yung trypsin and collagenase. Itong trypsin and collagenase, sila yung pinakamakikita sa reagents na ginagamit para ma-disrupt yung extracellular matrix ng tissues. So, di ba, kunyari, meron kang isang buong tissue. Yung gagawin mo, kailangan mong tanggalin yung pagkakadikit-dikit nila. Para matanggal yung pagkakadikit-dikit nila, lalagyan mo sila ng proteolytic enzymes. And specific proteolytic enzymes na ginagamit ay itong trypsin and collagenase. And another chemical use is yung EDTA. Na-encounter nyo na to doon sa DNA extraction natin, di ba? EDTA or ethylene diamine tetraacetate yung ends on which yung cell-cell adhesion natin, yung pagkakadikit-dikit ng cells natin, they are dependent on calcium ions. So therefore, kailangan mo tanggalin yung calcium ions. Paano mo matatanggal yung calcium ions? Mag-i-introduce ka ngayon ng EDTA. Kasi yung EDTA, kaya niyang mag-bind doon sa calcium ions. And kapag nakabind na siya doon sa calcium ions, hindi na yun magagamit ng cells natin. Ayun. So, using these enzymes and these chemicals, you can now disrupt the extracellular matrix and cell-cell junction of your tissues. So basically, in simpler terms lang, tinatanggal mo yung pagkakadikit-dikit ng cells gamit itong enzymes na to and yung chemicals na to. So ayun. Kapag natanggal mo na, ito pala yung picture ng EDTA. So ito yung picture ng EDTA and trypsin. Ganito actually yung makikita mo sa laboratory, sa cell culture laboratory. Ito yung itsura ng trypsin EDTA solution. Magkasama na yung trypsin and yung EDTA. Yan yung bago sila i-cell culture. Ayun. Next is you have to separate the cells in cell types from a mixed cell suspension. If yung tissues na ginamit mo ay mixed yung tissues na ginamit mo. So for example, kunyari kumuha ka ng blood. So yung blood, 'di ba, iba-iba naman yung cell types doon. Kailangan mo pa i-separate yung specific cell na kailangan mo doon, 'di ba? So kailangan mo muna siya sort out. Yung isang technique na ginagamit ay yung tinatawag na fluorescence activated cell sorter. Ano yung nasa fluorescence activated cell sorter? Simple lang. Kailangan mo lang ng yung cells mo, ilalagay mo dito. Tapos, lalagyan mo ng fluorescence yung cells na yon. And kapag tumama sila sa laser, magkakaroon ng charge yung cells na yon. And depende sa charge nila kung saan sila makakapunta. So, merong different cell collectors dito sa baba. Dito, masi-separate yung different types of cells mo depende dun sa naging charge ng cells. Ito yung parang automated way to sort out cells. Pero, in the laboratory rin, pwede ka rin mag-manual na isi-separate mo yung cells mo from other cells. So, yung pinakamadali ay yung sa blood, di ba? Yung sa blood, madali mo lang masi-separate yung red blood cells from your leukocytes or from your white blood cells or your plasma rin kapag mag-centrifuge ka. Ganon. Pero kung mas mahirap naman siyang i-separate, pwede ka gumamit ng fluorescence activated cell sorter. Ayun. Now, your cells, they can be grown in a culture. So, how? As in, nasabi natin yung term, excuse me, sinisipon ako, sorry na. So, yung term na in vitro, meaning in glass. So, yung cells natin, they are grown in vitro. In contrast to in vivo, sa loob ng living organism. Pero yung cell culture natin, in vitro natin sila ginagawa kasi sa glass. And itong pictures na to, yan lang rin naman yung examples ng cells na ginogrow in vitro or ginagawang cell culture. So, yung nauna ay mouse fibroblast, yung pangalawa ay myoblast, and ito ay retinal ganglial cells. So, ayan. Examples lang yan nung mga cells na pwedeng i-grow in your culture. So, makikita nyo, you can grow different types of cells in your culture. Kahit anong ma-imagine nyo na cells, pwede nyo sila ipag-grow in a culture. And magkakaiba-iba rin, meron din variations kung paano sila mag-grow. Pero yung titingnan natin ay yung parang pinaka-basic way on how to grow these cells. Yan. So, meron tayong tinatawag na growth conditions, dalawang types. 'Di ba? Kasi magkakaiba rin yung types ng cells na pwede nating i-grow. So, yung first ay yung nagka-create ng monolayers. So, in a Petri plate or in a Petri dish, or minsan rin sa flask, monolayers yung type ng growth condition. And makikita to sa adherent cell cultures. Adherent meaning they have to adhere to a surface. Kailangan nila kumapit sa surface bago sila mag-grow, di ba? So, ayun. And yung ginagamit na growth medium dito ay DMEM yung tinatawag. DMEM growth medium yung ginagamit na growth medium sa adherent culture cells, adherent cell culture. Yung isang type naman ng growth condition ay free-floating. And dito naman, nakasuspend lang yung cells mo doon sa medium mo. So, nagfo-float-float lang sila doon sa medium mo. And yung type ng growth medium na ginagamit ay yung RPMI. And ito yung ginagamit sa pag-culture ng white blood cells ng leukocytes kapag magka-karyotyping. Makikita niyo mamaya. And meron tayong tinatawag na culture media. So, di ba, kailangan mo ng media para mag-grow ng cells. So, yung culture media, ganito yung itsura. Ito specifically ay RPMI, ginagamit sa mga cells na free-floating. So, kulay red sila, yung usual na kulay nila. And yung laman nila, merong amino acids, merong inorganic salts, at merong vitamins. And these are the needed molecules for your cells to grow. Yung culture media mo naman, hindi ko pala napakita. So, wait lang. Yung culture media mo, pwede mo siya ilagay in different kinds of lalagyan. So, pwede ka magpa-grow ng cells dito sa flask, pwede rin sa plates, tapos meron din dito maliliit na plates. Depende kung anong type ng cells yung pinapa-grow mo. Yung nakita ko pa lang, actually, ay itong sa flask at itong sa Petri plates. Usually, ganitong types ng flasks yung ginagamit. And yung flasks dito, either cup sila na wala talagang butas or merong butas. Wait lang. Ayan. Next. Yung culture medium, they also contain this so-called fetal bovine serum. So, fetal bovine serum, alam niyo ba kung saan kinukuha? Sa batang cows. Kasi fetal bovine, di ba? Sa batang cows, sa baby cows, kinukuha itong serum na to. Di ba? Ang weird. So, kung ano pinagagawa ng scientists natin. They are collected from bovine fetus. And they give additional supplement to the media by providing the needed macromolecules, lipids, and growth factors na kailangan ng cells. Yung exact composition niya is unknown. So, for every gantong flask or for every gantong bottle, magkaiba yung composition niya sa isa pang bottle depende sa lot number. Alam niyo ba yung meaning ng lot number sa mga reagents natin? Yung lot numbers natin, yun yung parang nagsispecify kung ano kung ano yung batch na kung kailan siya pinuduce or ano yung batch na pinuduce siya. Parang specific siya sa kada bote ng reagents. And kailangan mo tingnan yung lot number minsan kapag mag-experiment ka para makita mo kung halos pareho yung laman ng reagents mo. So, mas maganda kapag balang araw mag-experiment kayo using these kinds of reagents, titingnan nyo yung lot numbers nila. And yung lot numbers na yon, i-compare nyo kung pareho. Hahanapin nyo yung magkaparehong lot numbers and yung magkaparehong lot numbers, sila yung pareho rin yung compositions. So, ayun. Pero sa fetal bovine serum, yung magkakaibang lot numbers nila, magkakaiba rin yung exact composition nila. Ano yung sabihin ko dapat? So, itong lot numbers na to, parang ano lang siya, yung sa batch kung kailan ginawa or parang batch ng paggawa ng reagents na yon. And yung reagents natin, halos the same lang yung mga laman nila. Pero mas maganda pa rin kung i-check mo yung lot numbers nila para makasiguro ka na halos na pareho talaga yung composition ng reagents na gagamitin mo kapag mag-experiment ka. So, ayun. Itong fetal bovine serum, sa different lot numbers, halos magkakaiba rin yung exact composition nila. Therefore, it can affect your cultures. Kasi di mo alam kung gaano karaming macromolecules, di mo rin alam kung gaano karaming lipids or growth factors yung nandoon sa fetal bovine serum mo. Parang in-extract lang siya ng mga scientists natin, tapos nilagay siya sa bote, ganun. And kailangan siya to grow your cell culture. But you have an alternative. You can use a defined media. Itong types ng media na to, alam na kung ano yung exact composition ng mga nung macromolecules, ng lipids, ng growth factors doon sa media na yon. Kaya mas even din or parang masasabi mo na even yung pagbagrow mo nung cell culture mo. And you also have to make use of a buffering system. Hi. So, buffering system is needed to ensure that proper pH is maintained. Bakit kailangan ng proper pH sa cell culture? Kasi, kailangan mo ng proper pH para proper din yung pag-grow nila. Kailangan mong i-replicate yung pH nung nasa katawan mo doon sa pH ng cells na nasa culture mo. Para yung growth nila halos under normal conditions na kung paano sila nag-grow kapag nasa loob sila ng katawan. Diba? So, yung buffer system na ginagamit ay particularly yung sodium bicarbonate. It keeps the pH of your cell culture between 7.2 and 7.4 with 5 to 10% gaseous carbon dioxide. And ito yung normal pH sa cells natin sa katawan. Or normal pH ng halos lahat ng cells sa katawan. And you also have to use a pH indicator. Yung ginagamit ay yung phenol red. Kaya makikita nyo, halos lahat ng medium natin ay phenol red to visually monitor the change in pH. Kasi delikado kung magbago na yung pH mo, ma-affect rin yung cell culture mo. So, minsan yung cells natin, kapag sobrang acidic na, nagigisa lang, yung cell culture mo magiging yellow and scary na yon kasi ibig sabihin, malapit na mamatay yung cells mo doon sa cell culture mo. So, kailangan mo ng pH indicator. And aside from a buffering system, ano pa sa tingin nyo yung nakaka-affect sa cell culture natin? Ano pa kaya? Na halos related sa pH. Ang isa pang nakaka-affect sa cell culture natin ay yung temperature. So, kailangan mo rin i-maintain yung cell culture mo at a certain temperature. And ano yung normal temperature ng katawan natin? Ng katawan ng halos mammalians, di ba? 37 degrees Celsius. So, you have to maintain also your cell culture at 37 degrees Celsius. Pero kung ibang animal cells naman yung gagamitin mo, for example, magse-cell culture ka ng galing sa palaka, iba rin naman yung temperature na gagamitin mo doon. And iba rin yung buffering system na gagamitin mo doon. Pero, since halos mammalian cells lang naman yung ginagamit natin sa medicine to investigate or to research, ito yung kailangan natin i-maintain. And, yan, titingnan natin yung laboratory setup. And, nakita niyo na kanina, ganyan lang rin naman. Ano ba? Ganito yung itsura sa flow hood. Under flow hood ka gagawa, obviously, ng experiment mo para yung yung airflow ay controlled and under sterile conditions ka magsi-cell culture. So, meron ka ditong ethanol spray, meron ka ditong waste, dito yung culture flasks mo, tube rack, reagents mo, tapos yung pipetors mo. Ganyan yung usual na itsura ng cell culture laboratory. And bakit mo kailangan gumamit under the laminar flow hood? Kasi minsan nagkakaroon ng contamination. At ayaw natin magkaroon ng contaminations yung cell culture natin kasi sayang nung efforts natin kapag nagkaroon ng contamination. Example ng contamination ay yung bacteria. So, ito, as you can see here, ito yung cells natin. And kapag nag-zoom in ka dito, may makikita kang, wow, ang daming bacteria. So, kapag ganyan, delikado na yung cell culture mo. Dahil yung bacteria, they can affect the pH of your culture. And kaya maapektuhan nila yung pH ng culture mo and in turn, pwedeng mamatay yung cell culture mo. So, uulit ka naman from the start. Masasayang yung efforts mo. Another is yung yeast. So, nakita nyo ba? Basta may bilog dyan. Halos mas makikita mo sila as compared to bacteria. And, ayun. Kaya rin nilang mag-contaminate ng cell culture mo. Aside from yeast, yung fungi din, kaya rin nila mag-contaminate ng cell culture mo. Another is yung mycoplasma. And ito yung maliliit na bacteria, di ba? As you can see in the figure, may mga mycoplasma dito. Maliliit sila na bacteria and maapektuhan rin nila yung cell cultures mo. Pwede nilang baguhin yung pH. In turn, mamamatay yung cells mo. So, kailangan mong tingnan kung merong contamination yung cells mo. And also, isa rin na pwedeng mag-contaminate ay yung viruses. And delikado yung viruses kasi hindi mo sila makikita under the microscope. So kapag na-contaminate ang viruses yung cell culture mo, di mo sila makikita under ng microscope. Makikita mo na lang, bigla na lang mamamatay yung cell culture mo nang di mo alam kung anong nangyari. So paano ba natin ma-check kung merong contamination? So obviously, number one, kailangan mong tingnan under the microscope yung kung may makikita kang contamination and makikita mo yung bacteria, yung yeast, and yung mycoplasma, fungi din, kaya silang makita under the microscope. Pero yung viruses, kailangan mong kumuha ng sequence nung cells mo. So, kailangan mo pang ipa-sequence yung cells mo at titingnan mo kung merong pagkakaiba doon sa sequences mo. Yung usually na titingnan ay yung tinatawag na short tandem repeats or STRs. Yun yung titingnan kung merong ng contaminations yung cell lines mo. Ayun. Eh, ano yung gagawin mo kapag na-contaminate na yung ano yung gagawin mo kapag na-contaminate na yung cell lines mo? Kailangan mo ulit mag-culture from the start. So, kukuha ka doon sa unang pinagkunan mo nung cell lines mo, yung primary cells mo. Tapos, dapat i-make sure mo na wala ka ng contamination. And, ayun. So, ano pa bang sabihin ko? So, ayun lang. So, ito, manonood tayo ng video tungkol sa cell culture. Yay! Lang. Wala na. Meron ba kayong tanong so far? Okay lang yung discussion natin actually. Yung basics lang ng cell culture. Kasi baka balang araw may chance kayo na mag-cell culture, di ba? Kunyari, wait lang. Ano nangyari? Gusto niyo mag-cancer research, ganon. Wait. Ayun. Ito muna. Nakita niyo ba?
Yes po.
Okay. Sabihin niyo kapag naririnig niyo or hindi. Wait, i-play ko.
Culture depends upon many factors, including the quality of the cells and reagents you use, your sterile technique, the amount of experience you have, and the right treatment. Proper setup is important. Have everything you need close by. The basic equipment you'll find in a cell culture lab includes a cell culture hood, an incubator, a water bath, a centrifuge, a refrigerator freezer, a hemocytometer, and a microscope. You'll also need cell culture flasks, medium and other reagents, pipettes, and waste containers. The hood provides a sterile environment to work with your cells without fear of contamination. Always open your cell culture dishes and reagent bottles in a sterile environment. Outside the cell culture hood, keep a stack of disposable pipettes nearby. Pipettes are placed in the hood during use, only opened inside the sterile field. Have the trash, biohazard bin, and sharps container close. A cell counting machine, such as the Countess, gives you quick, accurate counts needed during culturing, or you can count cells manually with a hemocytometer. You may also wish to have the centrifuge located in the cell culture facility. Incubators provide the appropriate environment for cell growth. Carbon dioxide and temperature settings depend on cell type and medium selection. Mammalian cells are cultured at 37 degrees Celsius in the presence of 5 to 7% carbon dioxide with high humidity. Inside the incubator, keep your cell culture flasks evenly spaced and avoid crowding to ensure that the air inside the incubator moves freely. This helps to maintain temperature, humidity, and proper gas exchange. Don't make very tall stacks of plates. They may fall over, and lower ones may not get the appropriate gas exchange. Store all media and reagents according to the instructions on the label. Some components are known to degrade when left at room temperature or exposed to light for extended times. If the medium will be stored in a glass front refrigerator or a cold room, wrap the bottles in tinfoil or store them inside a box. Some labs warm their reagents to 37 degrees Celsius before use. Warming reagents should take 10 to 20 minutes, depending upon the size of bottles you are using. Others choose to use reagents warmed to room temperature or directly from 4 degrees storage. If you use a water bath, make sure to keep bottles upright with necks out of the water. Specially designed bottle weights are available for use with the Gibco bottle. Alternatively, metallic bath beads can be used instead of water. Bath beads are a cleaner, greener alternative to the water bath. While waiting for reagents to the materials you'll need to use. When working under the hood, make sure the hood sash is at the proper height. Correct chair height and body position prevent shoulder stress and also helps prevent contamination. Never breathe or talk into the hood. Wipe the hood with 70% ethanol before and after use. Each item in the hood should be sprayed with 70% ethanol and wiped clean. Items within the hood are usually placed as follows: pipette aid in front, reagents in back, tube rack in the rear left, small container to hold liquid waste in the back right, your cell plates in the center. Never leave reagents in the water bath longer than necessary. Excessive warming can lead to L-glutamine degradation and loss of activity in trypsin solutions. Gibco Glutamax and Triple are more stable versions of L-glutamine and trypsin. Remember to clean each item placed in the hood with ethanol. You may choose to spray the item before placing it in the hood or immediately after, as demonstrated. Transport your cells carefully. Don't allow damage to the cell monolayer and avoid medium splashing that can encourage contamination. If using flasks with unvented caps, be sure to tighten before removing from the incubator. View every culture under the microscope daily. Check on cell health and growth and look for contamination. Make sure you're working with the cells you are expecting in your culture. If there's any doubt about cell identity, get a new culture from a reputable source or use a cell authentication kit. Accurate count. Keep a stack as a sterile environment.
So, ayun. Hanggang video lang tayo para ma-feel nyo kung ano yung loob ng isang cell culture laboratory. So, sa St. Luke's din, halos ganyan lang rin yung setup namin kapag nagpo-culture kami ng white blood cells pag magka-karyotyping. And makikita nyo, nasa loob, i-prepare mo yung reagents mo, yung mga red dito. And kung napansin nyo man, yung mga cell cultures natin, nandun sila sa parang flasks, di ba? And yung sinasabi ko kanina, yung flasks na yun, may pagkakaiba sa caps nila. Wait lang, hanapin ko. Saan na yun yung flask? Ayun. Itong flask, nakikita nyo ba? Hello? Nandyan pa ba kayo? Kung nakikita nyo man, yung flasks, yung caps nila, magkakaiba yan, depende sa cells na i-culture mo. So, sa situation na to, yung caps dito, merong onting butas, tapos merong filter. Kasi, kailangan na makapasok yung carbon dioxide sa loob ng flasks para matulungan na mag-grow yung culture. But some of the flasks actually, wala silang butas and wala silang filter. Yung mga flasks na yon ay ginagamit sa cells na hindi kailangan ng carbon dioxide or ayaw mong malagyan ng carbon dioxide yung cell culture mo. So, ayun lang. Ano pa ba? Ayun. Sa next video naman, ipapakita natin kung paano mag-passage ng cells or kung paano mag-transfer ng cells from one media to another. And kailangan matutunan yung technique na to kasi crucial siya sa gawa ng cell lines or pag-maintain ng cell lines. Dahil halos araw-araw kailangan mo i-check yung cell lines mo kung maayos pa ba sila. And kapag at a certain time din, kunyari yung schedule mo na mag-transfer or mag-passage ng cells mo, kunyari Monday and Thursday, kailangan mong gawin ng maayos yung passaging kasi dun nakasalalay yung future ng cell lines na yun. So, for example, yung HeLa cell lines, kung hindi maganda yung pagkapag-passage nung cells na yun, hindi na rin ma-maintain yung integrity ng cell line natin. So,
The growth of cells in culture follows a standard pattern. A leg after seeding is followed by a period of exponential growth called the log phase. Cells should be passaged when they cover the plate, or the cell density exceeds the capacity of the medium. Maintaining log phase growth will maximize the number of healthy cells for your experiment. Before retrieving your flasks from the incubator, sterilize the hood and have all required supplies at hand. Examine your culture carefully for signs of contamination or deterioration. Handle the cells with care during transport. For adherent cells, remove the spent medium from the flask using a sterile pipette. Rinse the cells with a balanced salt solution such as DPPS. Make sure to use a salt solution without calcium and magnesium, as these may inhibit your cell dissociation reagent. After rinsing the cells, remove the salt solution. Each time you aspirate liquid off the cells, place the next solution on quickly. Add your cell dissociation reagent to remove cells from the plate. Use just enough solution to cover the cell sheet. Consider using a gentle dissociation reagent, such as Triple Express, to avoid damaging your cells during dissociation. Make sure that the solution completely covers the cells. You may want to tap gently on the plate to help the cells detach. Use a microscope to confirm the cells have released from the flask. They will start to appear round as they release from the substrate and will move or slide when the flask is tilted. Complete dissociation is necessary. Do not leave the dissociation reagent on too long, especially if you're using a reagent other than triple. You may choose to manually break up lingering clumps by repeatedly pipetting warmed medium over them. A single cell suspension is important to achieve an accurate cell count. If you are using trypsin, the collection medium will need serum, or you'll need to use a trypsin inhibitor to inactivate the trypsin. If you're using triple, inactivation is achieved by dilution alone. No serum is needed. Transfer the cells to a conical tube and centrifuge to remove any residual dissociation reagent. The speed and time of centrifugation will vary based on your cell type. After centrifugation, you should have a well-formed pellet. Remove the medium from the centrifuge tube with a pipette and discard the medium into a waste container. Try not to disturb the pellet. Resuspend the pellet with warm, complete growth medium. Gentle pipetting will disperse the cells to ensure a homogeneous solution of single cells. Remove a small sample for cell counting. Trypan blue is used when counting cells to indicate the ratio of live to dead cells. The stain turns dead cells blue, but healthy cells with an intact membrane will remain white or colorless. Using the microscope, count the total number of cells and the number of dead or blue cells. Based on your cell count, determine how much additional fresh medium to add for optimal seeding density. Add the required medium, mix the cells gently, and pipette the solution into fresh flasks. Cap the vented caps on your flask tightly. If the caps are not vented, cap loosely. This keeps contamination out while allowing adequate gas exchange. Use a north-south, east-west motion to evenly distribute the cells and transport the flasks to the incubator. When passaging suspension cells, you'll begin by removing a small sample from the cell culture flask for counting. You'll follow the same counting procedure for adherent cells using Trypan Blue and either a hemocytometer or the Countess automated cell counter. Based on your cell count, add additional fresh medium to the flasks. stay within the minimum and maximum volumes. This is important to maintain optimal air exchange and shaking flow. You may need to split the culture into multiple flasks. Cap the flasks appropriately, depending upon whether they are vented or not. Return them to the incubator. Every 3 weeks, gently spin the cells from every new medium. This eliminates cell debris and metabolic waste.
So, ayun. Hindi nyo naman kailangan i-memorize yung steps. Pinakita ko lang sa inyo para ma-feel nyo kung ano talaga yung actual na nangyayari sa isang cell culture laboratory. Ang kailangan nyo lang tandaan ay yung mga reagents na ginagamit at kung ano yung functions ng kada reagents na yon. Ayun. Wait lang. Cell culture. Ano tayo? Meron ba kayong questions so far? So, ayun. Aside from culturing cells ng paggawa ng cell lines, you can also make hybrid cells. So, una, bago natin ito i-discuss, bakit ka gagawa ng hybrid cells? Bakit anong trip mo sa buhay at bigla kang gagawa ng hybrid cells? Itong hybrid cells, also called your hybridomas, they are used to produce monoclonal antibodies. So, if you can produce monoclonal antibodies, pwede ka rin makagawa ng antibodies. Pwede kang gumawa ng gamot sa certain types of diseases. So, naimbento tong paggawa ng hybrid cells through cell culture dahil minsan mahirap mag-isolate ng antibodies galing sa katawan mismo kasi ang dami-daming antibodies, ang dami-daming antigen. Ang hirap na i-sort out mo pa yung antigen, ang hirap na i-sort out mo pa yung antibody na kailangan mo or kailangan mong i-investigate. So, ngayon, merong technique na ginagamit to produce hybrid cells through cell culture. And yung hybrid cells na to, kung ano lang yung target antibody na gusto mong i-produce, yun lang rin yung mapoproduce niya, di ba? Monoclonal antibodies yung pinoproduce. So first, magsususpend ka ng 2 cell types. And in centrifuge mo sila gamit ang fusing agent. So dito naman, yung gagawin mo, if-fuse mo itong isang differentiated normal cell sa isang mouse tumor cell. Usually, ito yung ginagamit. Tumor cells from mice. And kapag na-centrifuge mo na sila, magkakaroon ng cell fusion and formation of heterokaryons which are then cultured. Wait lang, laser pointer. Heterokaryon, yung tawag dito, kapag na-fuse mo na yung dalawang cells na gagamitin mo, magkakaroon ka ng heterokaryon. And sa heterokaryon, kung makikita nyo, meron ka pang dalawang nucleus dito. Meron ka pang dalawang nucleus dito, so hindi pa sila talaga fused. Para makapag-select ka ng cells containing fused nucleus, you have to let them grow in a selective medium which allows only heterokaryons to survive and proliferate. These become hybrid cells which are then cloned. So, i-grow mo sa isang selective medium itong heterokaryon, and dito mo makikita kung alin yung cells doon yung nag-fuse yung nucleus nila. At makakagawa ka ngayon ng hybrid cells. And itong hybrid cells na to, they can produce monoclonal antibodies. So, kung may target kang antigen, you can use these hybrid cells to produce your target monoclonal antibodies din. So, pwede ka ngayon manggamot ng mga certain kinds of diseases. So, example, ano bang pwedeng example natin? Kunyari, gusto mo gamutin ang dengue. So, ngayon, kung gusto mo gamutin ang dengue, pwede ka rin gumawa ng antibody para sa dengue. Gagawin mo, kukuha ka ng antigen doon sa dengue na yon and magpo-produce ka ng antibody para sa dengue na yon. And para makapag-produce ka ng specific monoclonal antibody para sa dengue, kailangan mo siyang i-incorporate yung antigen dito sa cells na i-fuse mo. Kapag na-incorporate mo na yung antigen dito sa cells na i-fuse mo, pwede ka na ngayon gumawa ng hybrid cell na makakagawa ng antibody against that antigen. For example, against dengue. Ayun. May tanong ba dito? Ito na yung last. So, other example application. Other example applications ng cell culture, dito na yung karyotyping. Wala akong picture dito actually. Kasi di ako nagpipicture ng lab works namin. Sayang. Pero bago ka magkapag-karyotyping, kailangan mo muna i-cell culture. Alin sa tingin nyo sa blood natin yung ginagamit para mag-cell culture? Di ba merong RBC, merong white blood cells, meron din yung plasma, alin doon sa hula nyo yung ginagamit para mag-cell culture and magamit sa karyotyping? Meron bang gusto mong hula? Or hulaan nyo na lang, alin kayo yung gagamitin natin? Red blood cell or white blood cell? Para kapag-karyotyping? Or para ma-culture natin? Red blood cell kayo or white blood cell? Meron ba gusto mong hula? Wala? Walang gustong manghula? Wait lang, meron pa lang nagta-type. Sorry, di ko nakita. White blood cell. Tama. So, sabi ni Mr. Cleofe, ni Mr. Cuba, ni Arcilia, and ni Ms. Bagaporo, white blood cell daw. Tama. Bakit? Kasi white blood cell lang yung merong nucleus. Therefore, merong genomic material sa blood natin. Yung RBC, di ba, enucleated sila. Enucleated meaning wala silang nucleus. So, hindi ka naman makakapag-karyotyping gamit yung red blood cells mo kasi wala silang nucleus. Ano yung makukuha mo doon, di ba? So, yung gagamitin natin ay yung white blood cells. And in karyotyping, yun yung kinukuha. And essential na ma-cell culture mo yung white blood cells ng maayos para makakuha ka rin ng magandang karyotype. So, nakasalalay rin sa pag-cell culture mo ng white blood cells yung magiging karyotype mo. And ito rin yung ginagamit sa detection ng chronic myelogenous leukemia. Ano yung naaalala nyo sa chronic myelogenous leukemia? Diba merong BCR-ABL. Nagkaroon ng translocation yung chromosomes natin, yung from 9 to 22. Tapos yung ABL gene mo, nag-fuse siya doon sa BCR gene mo. So sa karyotype, makikita mo ngayon kung merong chronic myelogenous leukemia yung patient mo. And makikita mo yon gamit yung white blood cells na kinulture mo. So yun yung application na natin ng cell culture sa under laboratory setting na pwede siyang gamitin na pag-detect or pag-detect ng sakit. So, example natin doon ay chronic myelogenous leukemia. Okay, walang picture. So, ayun. So, tapos na. In summary, we learned about how cell culture came about and we also discussed about cell culture growth conditions and the laboratory setup. Hindi ko na-type, pero nakita nyo rin, pinakita ko yung paggawa ng hybridoma and its use to produce monoclonal antibodies. And also, yung application niya ay isang application ng cell culture din ay sa karyotyping. Bago ka makapag-karyotype, kailangan mo munang i-cell culture yung white blood cells mo and makita mo na yung karyotype ng tao. And also, yung karyotype na yon pwede ginagamit rin sa pag-detect ng certain diseases such as your chronic myelogenous leukemia. Yan lang. So, yan lang. Meron ba kayong tanong sa lesson natin this afternoon? Meron ba? Meron ba? Maikli lang yung lecture natin as compared sa mga dati. Meron ba kayong tanong? Hello? Nandyan pa ba kayo? Hello, hep hep? Hep hep? Walang nag-uhuri? Walang mananalo? Okay. Hep hep? Okay, nanalo si Shania. Congratulations. Panalo ka ng bragging rights. lang. Wala akong mapapadala. Wala na ba kayong tanong? Okay. Kung wala ng tanong, thank you for listening to today's discussion. Sana naman may natutunan kayo bago. Kung may tanong kayo, i-chat nyo lang ako. At, ayun lang. Bigyan nyo na sa akin yung code names nyo. Thank you. Goodbye. Stay safe, guys. Bye. Bye na.
Thank you, ma'am.
Thank you po.
Thank you po.
Po.